Hello and welcome to Psychology 301. This is the lecture for Unit 5. It's lecture E. And we're talking about animal cognition, but we're going to focus on timing and on order. So let's talk a little bit about the role of timing. Throughout the quarter, we've mentioned repeatedly the importance of time as it relates to learning and behavior. The one constant in our environment is time. Time influences performance in almost every single type of behavioral learning or behavioral mechanism that we've reviewed. Time has been important in habituation and spontaneous recovery. Habituation only occurs with repeated exposures over time, and spontaneous recovery occurs with the passage of time without simultaneous exposure to the stimulus. So time plays an important role both in the establishment of habituation and the recovery of a habituated response through, for example, rest. Classical conditioning, we saw a very important role of timing and temporal contiguity, very important for classical conditioning to be successful. In operant conditioning, we saw that the effect of delaying a reinforcer or a punisher can decrease its efficacy in terms of influencing behavior. Delays reduce punishment efficacy. And in long-term stimulus control, we're really talking about memory is really nothing more than stimulus control over time in the absence of a particular stimulus. So delays, if they're long enough, can impede memory. So we know that time influences are how we go about learning and the processes that we engage in when we are learning. With animals, we've seen evidence of an animal's ability to keep track of time. We see auto-shaping, which is the gradual development of a particular behavior over time. When we look at fixed interval responding, we see scallops that indicate sensitivity to time. We see that when there are cues for behavior, but they're not signaled, we get differential discrimination. So if we don't have cues, we have to remain, the only cue available is time. And when that's the case, sensitivity to time allows for learning to occur. And of course, we have unsignaled avoidance conditioning. Even though an organism doesn't actually know when a particular aversive stimulus is going to be presented, if it's presented on a fixed interval, they're able to learn and engage in avoidant behavior. So to demonstrate timing, to really directly demonstrate timing, we have to rule out all the other possible mechanisms or other solutions that might be available to an organism. And when we do that, when all that's left is time, then we're able to definitively say that sensitivity to time is a critical feature in a particular type of learning. The author of your book, Michael Domshan, said, and I quote, "An animal is considered to be timing if the duration of event serves as a discriminative stimulus for them." In other words, if the passage of time is reliable enough, time itself can be an SD or an S plus for behavior. And there are several ways to directly investigate the ability to keep track of time. And the two of them that I want to talk about are the duration estimation procedure and the peak procedure. In the duration estimation procedure, rats are presented with two auditory signals that are identical but differ in duration. So you have a tone that's 5 seconds long and a same tone that's 8 seconds long. And there is a very brief half-second retention interval. And after the retention interval, two levers are inserted into the operant chamber. And the rules for the rat are, if the tone was a 5-second tone, press the left lever. And if the tone is a 8-second tone, then press the right lever. It's that simple. It's counterbalanced for rats. So half the rats learn 5 on the left, 8 on the right. The other half learn 8 on the left and 5 on the right to counterbalance for side. Correct matches produce food. Errors produce brief blackouts. And the results are really simple. The rats learn it. And they learn it very well as long as the retention interval isn't too long. So a half second, 1 second, they do pretty well. Okay. But it's a different thing to ask if a rat can actually generate a pattern of behavior based on when reinforcers are arranged in time. So can rats predict when a reinforcer is expected to be delivered? Can they actually learn that and behave in a way that shows us they know when the reinforcer is coming? So in the peak procedure, which was conducted by Roberts in 1981, it's a very simple procedure also. In phase 1, the rats are trained to lever press on 2 different schedules, usually a fixed interval 20-second schedule and a fixed interval 40-second schedule. And they're each correlated with different stimuli, and they get extended training. They get 6-hour training sessions over 10 days. So they get about 60 hours of training on this, and they get really good at it. When stimulus A is on, it's a fixed interval schedule. When stimulus B is on, they respond accordingly. And both of these, because they're fixed interval schedules, produce fixed interval scallops. The FI 20-second schedule is smaller and shorter than the fixed interval 40-second schedule, but you get 2 different scallops for each of the 2 schedules. Then in phase 2, they test these 2 schedules, A and B, in extinction. They turn on the stimulus for A and let it run, and it runs beyond the 20-second time frame when reinforcement should be delivered. They do the same with stimulus B. They present stimulus B, and it is on longer than the 40 seconds that would be expected. And they look to see what the responding during extinction looks like. Let's see here. So this is the pattern that you see. You see a pattern of responding that shows, I'm going to use my cursor here for a moment, the 20-second schedule are the open circles here. The 40-second schedule are the dark squares. And if you look, you'll see that the peak in each of these 2 patterns of responding. So notice that early in the schedule here, there's very little responding. They start to increase over time. And as they begin to approach that 20-second mark, it tends to be at its highest level. And then as you go beyond 20 seconds, it starts to drop off until they don't respond anymore. The same is true with the 40-second schedule, except the peak is directly above the 40 seconds. So during testing, stimulus in A and B are presented for unusually long periods of time with no reinforcement and beyond the point when food would be expected. And what they're measuring is the rate of responding at different intervals in time. And what we find is that the animals, the highest rate of responding for both schedules occurs directly over the point when the reinforcer should have been delivered. And the data are extremely orderly, which suggests tremendous sensitivity to the arrangement of reinforcers in time and the expectations that these organisms, that these rats and these other animals develop based on these fixed temporal patterns. Weirden and McShane replicated these findings with humans and got exactly the same patterns. But what do these data mean? What does it really mean? There are some researchers, cognitive researchers, who believe that animals have some kind of an internal clock, which they consult unconsciously when they're timing. And the clock is thought to be part of the nervous system. So the physiologists have a say in this. The cognitive psychologists say, oh, it's an internal clock. The physiological psychologists say, well, it must be neural. This is a really good example of cognitive heuristics. Clearly, there isn't a Rolex or a Timex wrapped around the animal's brain. So there's not actually a timing device. You can't open up an organism's head and find the clock. All right? But the analogy of an internal clock could potentially be valuable in terms of the way we do research. So Russell Church is one of those people who has one foot in the behavioral camp and one foot in the cognitive camp. And he's one of the people interested in timing mechanisms. And his work shows that cognitive heuristics can help to stimulate and to organize and to direct the research efforts that are put forward to understanding this idea of temporal control of behavior. They can help stimulate questions in ways that might not arise otherwise. and the physiologist, a physiological organ that's responsible for timing, it might exist, and it's only likely to surface if we assume that it's there and if we have data that help direct us when looking for it. So there's a lot of value from the side of the cognitive psychologist or cognitive behavioral psychologist would argue that there's a lot of value in these cognitive heuristics because of what they do in terms of the way we think about the questions that we ask when we want to study timing. So for example, does the timing mechanism work like a clock? Or does it work like a metronome? Or maybe like a stopwatch? Or maybe even a sundial? Each method of timing is very different and have implications that are different for how the challenge of keeping track of time would be achieved. And so each one of these orientations makes us look at the idea of biological timing very differently. So Russell Church, this is his model. This is John Gibbon and Russell Church's information processing theory of timing. And this is right out of your book. There are three processes associated with this model. I'm going to use my cursor again here for a moment. So early on in the process, we have a clock process that's going to keep track of time per se, or keep track of paces or clicks, if you will. There's a memory process where we compare what we have accumulated thus far to what our memory is. And then there's a decision process where we decide whether or not to respond. So these three processes, let me go over each of them independently. The first one I want to go over is the clock process, which involves a pacemaker, a switch, and an accumulator. The clock process is initiated by the onset of a discriminative stimulus, a signal or a cue that time has begun, and usually it happens at the beginning of the interval. If we think of this as a fixed interval challenge, we could think of this in terms of it being maybe a fixed interval 20-second schedule. So the minute the SD comes on, the clock process begins. And the pacemaker at the beginning begins to generate pulses. You could think of them as clicks or as pulses or just some kind of regular pattern that the pacemaker is generating. And those pulses are fed into an accumulator that's adding them up. So, you know, there's, this is kind of like the brain going 1001, 1002, 1003, 1004. So it's accumulating pulses. And these pulses are fed into the accumulator. And when the accumulator reaches some target, a switch closes and a signal of information is sent to working memory. Okay? When it arrives in working memory, you have this current accumulation of pulses. And the organism also has reference memory, which is a past storage of previous experience that are similar to this one. So the animal has experience working in this fixed interval 20-second schedule for 6 hours a day for 10 days. For a 10-day period, you have 60 hours of experience. And so they bring forward the average representation of the amount of pulses required for reinforcement to occur, and it compares, it then shifts both the memory version and the current version to the decision-making process. And in the decision-making process, the pulses from the clock or from the pacemaker are compared to to reference memory, and they make a decision about whether they're equal to or greater than what's necessary from previous experiences. And if it is, respond. And if they're not sufficient, then you need to wait, open up all the switches, let some more pulses accumulate until you reach enough pulses to equal or surpass what you're expecting from previous memory. Now, this is a very cognitive model. It's very much an information processing model of timing. And if you open up a human brain, let's say we, zip off the skull and look down inside or do an MRI, you're not going to find one organ that looks like a Rolex. It's not generating pulses that can be heard or seen on an MRI, and they're not, and you're not going to see switches opening and closing. This is very much non-existent. These are analogies to what could be happening with the neurobiology of the brain, but the physiologist would have to resolve how that's done. Okay? Now, is there actually an independent clock? Well, what we really want is independent evidence of an internal clock. So, for example, some of the behaviors that have been shown that might actually be evidence of a clock are musical rhythm in humans. The fact that we can go 1001, 1002, 1003 in a pretty reliable pattern. When we're waiting for something and we have to wait a certain amount of time, we might tap our foot or tap our fingers. New moms will swing their babies back and forth on their hips. And when they're standing in line, even if they don't have babies, they might cross their arms and begin that same swinging motion. There's even what we call adjunctive behaviors in animals. When rats are solving time-related problems, they twitch their whiskers in kind of a rhythmic pattern. And pigeons will show an almost imperceptible fluttering or beating of their wings that are very rhythmic. And so it shows that they're able to actually, using something inside their body, they're able to generate these patterns of music or patterns of rhythms that could be related to this internal clock. Now, Peter Killeen and Gregor Fetterman proposed that, in fact, it works the other way around. They propose that the animals that are working on these time-based tasks are actually using these adjunctive behaviors as overt timing devices. So, for example, when we have to keep track of time, we say 1001, 1002, 1003. Are we keeping track of something inside of ourselves, or are we generating a pattern that we can then read in order to tell time? So the behavioral theory says that adjunctive behaviors are correlated with specific behavioral states like post-reinforcement behavior and search behavior. And as a result, there's no need to infer or assume complicated, unobservable cognitive processes that may not even be happening. That we can gain some degree of independence. You can actually videotape animals when they're working on fixed interval schedules and see whether or not their adjunctive behavior correlates with their responses. In the end, this entire system is limited. Nobody has developed a model that will allow us to distinguish between the Gibbon and Church cognitive heuristic model and the Killeen and Fetterman behavioral model. And in fact, the two of these models are almost a form of circular reasoning. And so it's a real challenge for us to resolve the question of timing. What we do know is we may not be really sure how they do it, but what we do know is that they remain sensitive to the passage of time, and they're able to use that as important information, even as discriminative stimuli for their behavior. And so there's still a lot of work left to do in this field of timing for us to understand exactly what's going on. Okay, now I want to talk a little bit about serial pattern learning. And I want to start by talking about order and organization in the world. And orderliness is really the hallmark of nature. It surrounds us. It's everywhere around us. It's outside of us. It's within us. From the tiniest chemicals and, oh gosh, that should say DNA. Let me fix it. I'm sorry about that. DNA, there we go. DNA structures to the organization of the universe. There's so much orderliness to life. And randomness is real. It does exist. And chaos is part of life. But order is very, very common and very, very important for learning. And our sensitivity to orderliness is reflected in how easy it is for us to learn about events or information or sequences when they're already organized. So we know that organization and order in the world is pervasive, and it facilitates our ability to learn. Let me give you some examples. We find both bilateral and radial symmetry in the world. This is order. Human beings, like the fly, are radially symmetrical. You could divide us right down the middle. Our brains have two hemispheres. We have two arms. We have two legs. We have two kidneys. Females have two ovaries. I mean, we have two of almost everything. And the things we only have one of often have two halves. So bilateral just means two halves. There's also something called radial symmetry, and we see it in things that are round, or here a sea star has radial symmetry. We see it in sea urchins. We see it in certain patterns in airflow. We even see it in hurricanes and tornadoes. You see that radial symmetry, those round patterns. We see order in fractals. We see order in the double helix of the DNA molecules. We see patterns and order in chemical compositions. This is actually generic Viagra, in case you were wondering. That's what Viagra looks like. Think about the way our cities are organized. Although these are generated by humans, they're generated in ways to make learning about the city easier. So Seattle on the left has a grid pattern, and the grids are very clear. They're numbered, and a lot of cities will have roads that run north-south or east-west, or run on the diagonal, so northwest to southeast and northeast to southwest. But on the right is Houston, Texas. And you'll notice that it's not really a grid pattern, but the freeway system is actually got some of that radial symmetry to it as well. They're concentric circles. There's a little tiny one in the very center, and then a little ways out there's another freeway, and then there's a third freeway that's outside of that. And that radial symmetry is also a way of organizing a city. Even our behavior is ordered. So this was kind of cool because I found this little flyer, and it says my ideal morning routine. I want to wake up, I want to open the blinds and make my bed, then I'm going to wash my face and fill up my 1-liter water bottle. I have no idea what blogilates are, but I think it's probably either writing about Pilates or reading about Pilates and then doing them. I'm not sure. It looks like exercise. 5 minutes of journaling, then read 10 pages of a book, then a 10-minute meditation, finish drinking that 1 liter of water, get dressed, put on your makeup, and have breakfast. And I suspect the woman who wrote this probably tries to follow that every morning. Now me, I'm like, get up, get dressed, take a shower, get dressed, brush my teeth, put on my makeup, get to school. I don't have a whole, make some coffee if I'm lucky. So my routine is a lot shorter and a lot less relaxing and peaceful than hers. But still, each one of us has our own pattern. I want you to see a video. So I want you to go to the PowerPoint presentation of this lecture and click through to this page, which is page 26. And I want you to click on where it says Japanese pufferfish. This pattern in the picture that you see, and actually I can show you where he is. This little fish, he's right here. He's right there. This is a courtship display that the male generates on his own. He creates this spectacular, you might call it a nest, but it's not even really a nest. It's just, it's a modification of his territory to attract females. And it's amazing. And the video here, which I will also put in the video section of this Japanese pufferfish, is narrated by David Attenborough, and it's amazing. And you'll actually get to see him generating and working on this. And the radial symmetry of this pattern is extraordinary. And it's for him, it's just a modal action pattern. That's how deeply ingrained this is genetically in his biology. It's powerful. Okay, so take a moment and go do that either now or after this video's over, and go watch the Japanese pufferfish because he's extraordinary. Okay, so excuse me. So serial order of events represents the simplest form of organization. In other words, the order in which you have to do things. And it makes a difference. It matters when we have to do things in a certain order, it actually makes it easier for us to learn. And we learn to do things in that order. So for example, have any of you ever, we all like to have orange juice in the morning and we all like to brush our teeth, but have you ever done them in the wrong order? Brushing your teeth and drinking orange juice is one of the most aversive things I can imagine in the morning. The flavor, that minty flavor of the toothpaste, makes the orange juice horrible. Some of you might even be kind of puckering while I say this because it's so unpleasant. The order matters. Look, I got another typo, sorry. Getting your car out of the garage. I hope to goodness that you do this in the right order every single time, and that you open the garage door before you put the car in reverse. So that's important. The order matters. Baking a cake, the order matters. You can't put the ingredients in the oven, bake them, and then mix them together. You have to do things in the right order. And the order that you do things at the ATM, it matters. You have to put in the right information. If you put the amount you want in when it asks for your PIN number, it's likely going to take your ATM card. So you have to be careful about order. So what is the role of serial learning, serial order in animal learning? Can animals, like a rat, isn't he adorable? He looks like Ratatouille. Anyway, can animals actually detect the order in a series of stimuli? Can they actually learn that stimuli are occurring in a particular order? Can they form a representation of that order so that they hold on to it in memory? Can they reliably reproduce that serial order? And even better, can they reliably generate their own unique serial patterns? All of these are interesting questions about what these little critters can do. So I want to start with one of the simplest studies, which is a study by Stuart Hulse and one of his students from 1975. And in this study, this is the runway maze study. It's very simple. The rats run down the runway. So they start in the start box, and they open the door, and there's a little tiny container, a little food well at the far right-hand side of this runway, and there's food in it. And in this study, Dr. Hulse manipulates how much food is in that little food cup. So he takes his little rats and he divides them into 3 groups. Group 1, there is a pattern to the way the food pellets are placed in that food well across the trials. So in trial 1, they run down, and usually these are like little sucrose tablets, so they're very, very yummy. The rats run down and they find 14 little tiny pellets. They're kind of like little BBs. And they find 14 of them. And then, oh, they're so good. And then he goes down after they finish eating them, and he picks the little rat up and takes him back to the start box and puts him back in and opens the door again. And he runs back down to the end of the runway. And this time there's only 7 pellets. And over the trials, it goes from 14 pellets to 7 to 3 to 1 to 0. And after that 0 pellet trial, it starts over. And it goes 14, 7, 3, 1, 0. And then it starts over again, 14, 7, 3, 1, 0. So it starts out with a large number and gets smaller as it goes. Group 2 also has a pattern, but in this case, the pattern is an increasing number of pellets. So it's exactly the same kind of trials as group 1, but they run in the opposite direction. So it goes from 0 to 1 to 3 to 7 to 14. And after you get 14, it drops to 0 and then starts over 1, 3, 7, 14, 0, 1, 3, 7, 14. Group 1 and group 2 have clear patterns associated with the number of pellets at the end of the runway on each trial. He has a third group, a control group, where they receive the same, all of the same numbers of pellets, the same number of 14 trials, 14 pellet trials, and the same number of 7 pellet trials, and 3 pellet trials, and 1 pellet trials, and 0 pellet trials, but they're arranged randomly. So there's no predictable order of what's coming next. All right. And what they do is he does this, and he puts the rats and he trains them over several days, and then he tests them, and the test is simply to measure how fast they run down the runway. That's it. It's really simple. And what we find is that groups 1 and 2 that have orderly patterns of reinforcement show very distinctive running patterns that suggest they're sensitive to the pattern of pellets. And group 3 doesn't do that. Group 1, that has pellets decreasing in size over trials, start out fast and run slower and slower and slower. Group 2, with the increasing food pattern, they run faster and faster and faster until they get to 14. And then for both groups, when they get to the 0 pellet trial, even though they haven't received 0 pellets till they get to the end, they go very slowly down the runway. They stop and they scratch their little ears and they sniff around, and those 0 pellet trials take forever. But the 14 pellet trials, they zoom down the runway. So there's a nice pattern to their running speed. Group 3 doesn't show any kind of pattern. They have an average speed. They tend to do the same thing on every trial because they have no idea what's at the end. So groups 1 and 2 have learned something that the rats in group 3 did not learn. It's related to the orderliness in which the pellets are delivered, and it directly influences their behavior. Now, the important thing is, we know they're sensitive. We can see that. They are learning something, but the question is, what did the rats learn? And there are several possibilities. The simplest possibility is just a simple association. Perhaps group 2 learns that 14 is always followed by 7, and that 7 is always followed by 3, and 3 is always followed by 1, and 1 is followed by 0, but 0 is followed by 14, and each one is a separate association. Another possibility is that they're learning more of an abstract rule, that the number of pellets decreases over time until there are none, and then it starts over. So they know they're getting a little bit less on each trial, and when it's 0, then it starts over. And that may be what they're learning, more of an abstract rule than just simple associations. And it's also possible that they get an overall structural analysis. They may learn that the sequence is 5 trials long. They may also know that there are always 3 pellets in the middle, and that the number of pellets on each trial nearly doubles with each successive trial, for example, for group 1. There may be a very detailed analysis. All 3 of these would lead to the behaviors that we observe, but we're not sure exactly what they've learned. So Herb Roitblatt, who's at the University of Hawaii, Manoa, and he studies rats and dolphins. He's a very cool guy. He tested his own rats' runway strategies by adding a misplaced 0 pellet trial right in the middle of the sequence. So they go from 14 to, normally they go 14, 7, 3, 1, 0. And what he does is, after they've learned that whole sequence on the testing days, he throws in a random 0 pellet trial. So it might be 14 on trial 1, trial 2 is 7, trial 3 should be 3, but it's 0 pellets. And then 3 and 1, and then 0 pellets. And then there might be another 0 pellets. And so he throws in on the testing trials these random 0 pellet trials. If the rats are learning a very simple association, then the odd 0 pellet trial should always disrupt their performance and reset them to whatever the next one should be following 0. So if the rats learn a simple association, the odd pellet trial should disrupt their performance, and the rats that always get 14 after 0 should run fast on the next trial. But if they have something more complex, like an abstract rule or an overall structure, then they should detect that the zero place trial is odd, disregard it, and run the next trial as if that zero trial had not occurred. And that's exactly what the animals do. The rat's performance is not disrupted at all. So they must be learning something more than a simple association. So let me give you an example of what they're really doing in a way that you can understand it. So we watch our favorite TV shows. One of my favorite TV shows right now, I had been a Bones fan, and right now I'm an NCIS New Orleans fan. And if someone were to ask me to relay the story of the last episode of NCIS New Orleans, I could do that. I could tell you the whole story from the beginning to the end and fill you in on all the details. But one of the things I wouldn't do is I wouldn't include the commercials. I would just ignore the commercials because they're not really part of the structure of the story. The rats are treating these zero pellet trials like commercials during their favorite TV show. They have this favorite activity, they've learned the pattern, and occasionally there's a weird glitch, and they treat it like a weird glitch. And they just continue on with their behavior. So it's very interesting that they're able to detect it as an error and disregard it. So there's something more than just a simple association going on. So Reuterblatt was not able to tell us what the rats are doing, but he was able to tell us that this behavior is not based solely on simple associations. Now, serial pattern learning and serial representation learning often are tied to behaviors that require smaller responses performed in a particular sequence before you get to the reinforcer. So things like tying our shoes or making the bed, or as I mentioned before, the ATM machine. These kinds of sequences are called response chains, and they don't receive the main reinforcer until you get to the very end of the chain. When you're at the ATM, you don't get your money until you get to the end of a very long sequence of behaviors that you have to engage in. And in a lot of cases with lab animals, these organisms don't even have to be aware of what the sequence is, because there might be an SD, there might be a discriminative stimulus that tells them what to do next. So at the ATM, for us, we put our card in and the card says, "Hello, Wendy Williams. What do you want to do?" And it gives us choices. Do you want to deposit? Do you want to take out money and withdraw money? Do you want to just look at your balance? And so there's a prompt that tells me what to do next. And there's a lot of prompting. So I push, I want to withdraw cash. And then it tells me to go, another screen comes up and it says, put in your PIN. And that means you have to put in the right number. So I have to do a long line of things before I get to my money, but each step is prompted. And that's true in the animal world too. Often there are prompts. But in some cases, there can be no prompts whatsoever. And the animals perform perfectly well. And we call that the learning the actual serial pattern. We call that serial representation learning. The animals have actually acquired an understanding of the sequence, and they've learned it well enough that they don't need to be prompted through the entire set of behaviors. So there's this great study by Mike D'Amato and Mike Colombo. I actually worked with Mike Colombo in New Zealand. Very nice guy. When he was in New Zealand, he was studying guinea pigs. But in 1988, he was studying Cebus monkeys. So they're like little capuchins. And these monkeys would come in and hop into their little operant chamber seat. It's kind of like a video game for them. And they often get juice as a reinforcer. So they sit down in their little chairs, and in front of them is an 8-key, what they call an intelligence panel. So instead of there just being 1 or 2 or 3 keys, there's 8. There's a line above and a line below of 4 keys each. And they're trained in this 8-key chamber to respond to a 5-sequence pattern. And in the article, he refers to it as an ABCDE pattern. They could be letters, literally letters projected on the screen, or it could be colors. Doesn't really matter. They could be shapes. It doesn't matter. But the important thing is that they have to do everything in the correct order. And so they call it an ABCDE series because they have to respond in that way. So when the keys come up here like this, you would have to go, I'm going to take my little cursor, you have to go A, B, C, D, and E. Okay? And on the next trial, it might be, it comes up again, the letters have moved, and now it's A, B, C, D, E. And as they continue on, the arrangement of the 5 keys they have to press changes from trial to trial, but they still have to do it in the right order. And if you do it in the right order, you get food or you get juice. And if it's an incorrect order, you get a 5-second blackout. The animals are trained extensively until they reach about 90% correct or better. And then they're put into a testing scenario. And in the testing scenario, they're tested with novel combinations, 2-item and 3-item subsets of the stimuli that they've seen before. And I've listed many of the possible combinations down here. So they're trained with AB only, AC only, AD, AE, BC, BD. And so here's an example. This is a 3-item set here, and they have to go B, C, and D. It's out of the center. That comes out of the middle of the sequence. It leaves off the A and leaves off the E. And so they're tested with these pieces, and they measure how long it takes them to actually start the sequence. They're measuring time from onset of the stimulus, the pattern, to the first response, okay? And they test them with these novel 2- and 3-item subsets where they run in order, and then they also test them with 2- and 3-item sequences where items are missing. So it might just be A and C, or it might be A, C, and D, or A, D, and E. And there are items in the middle that are missing. And they did both of these kinds of tests. And what they found was quite interesting. What they found was that, first of all, the monkeys perform well above chance on a normal sequence, and they do well above chance even on the tests. However, and that's true even with a 3-item series from the middle like B, C, and D, they still get it right. But what varies, the thing that is important in terms of the way their behavior changes, is that the latency to respond, which is how long it takes from the time the stimulus comes on until they pick the first item. So in this case, B, and then C, and then D. The time it takes them to get to B shows that if it starts with A, the A latencies are very short. If you get A, B, C, and that's all you get, they respond to the A immediately. But if all that's available is something other than A, so if it's the B or the C or the D that shows up as first in the list of what they need to respond to, there's a delay. So here we have, I'm going to use my cursor to kind of outline things. So here we have, in this one, this is the time to the first item is what this is. And we have 2-item sets are the gray bars and darker bars are 3-item sets. And what you find is if it starts with A, the latency is very short. But as you move progressively to the B, the C, and the D, the latency to respond gets longer and longer and longer. And when there are items missing, how well do they do? Again, depending on how many items are missing, they go, if there are no items missing, they're really fast. But as items in that pattern during the test are missing, they go slower and slower with each additional missing item. So, and Mike Colombo wrote in his paper that it seemed as though they were running through the entire sequence in their head before responding. So if the pattern is B, C, D, they look at the pattern, they realize that there's something missing, and he says it looks as though in their head they go, it's A, B, C, D, E. Where's the A? There is no A. Okay, where's the B? And then they respond. And so what they found is that it seems as though the animals are paying very close attention to the serial order, and when things are missing, they're using their own representation of that serial order to solve the problem. And as a result, their behavior changes based on how much of the sequence is missing. Very, very clever experiment. Okay, I'm going to stop there. This has been a long lecture. So I want you to take a break and go watch that video about the pufferfish and have a great evening. We'll talk to you later. Bye.